Good evening, everyone. My name is Takiwa Smith. I'm founder and executive director of Science, Engineering, Mathematics Link Incorporated, also known as SCM Link, and I want to welcome you to our Teen Science Cafe. Our Teen Science Cafe is a program of our Math and Science Career Academy whose goal is to expose you to STEM careers and engage them in hands on demonstrations and, in the time of virtual programming, interactive demos, but different ways to just introduce you to STEM professionals so you can explore STEM careers and learn about different STEM careers. Excuse me, my throat is so dry. Our Teen Science Cafe, our program specifically targeted to middle and high schoolers, provides you an opportunity to hear more in depth about a particular STEM career by meeting a STEM professional, graduate student, or a panel of those STEM professionals or grad students. So you can learn about their career journey, what they do every day, and get an idea of their field by them either doing a interactive demo, showing you videos or pictures of their work site or workplace, or talking about what they do every day. And the reason why we have this program focused on teens is because your teenage years are when you start making decisions about what you will do after you graduate from high school. So when you're in middle school, it helps you decide if you want to attend a STEM magnet school or what extracurricular STEM activities you may want to go to, such as robotics club, if you want to join any programs that any community organizations host, so you can get a little more STEM exposure. In high school, it allows you to do the same thing as far as pick what extracurricular STEM activities you're going to participate in, as well as deciding what your post graduation opportunities will be, whether you're going to apply to college, whether you're going to go to a community college or certificate program, but all of those things so you can plan your career starting at an earlier age where you can make decisions, hopefully because you saw someone in a field, in a STEM profession that you may be interested in, and decided that, hey, I listened to this person that was a speaker at the Teen Science Cafe. They are doing something that I am interested in doing, and they gave me the path to pursue that. And so, without further ado, I'm going to turn it over to Carlin Pounders, our program coordinator, who is going to introduce our speaker who will be here shortly. Um, she's had a little technical difficulties and she's resolving that. So, Carlin, I'm going to turn it over to you.
All right. Thank you so much, Tequila. So tonight we are going to hear from Dr. Shenell Tolson. He is a cell and molecular biologist. She works in human health and toxicology and uses her Instagram platform to inform the community about basic STEM and public health concepts, which we know recently that has been something that has been very important to do. So she is passionate about melanated munchkins in the STEM pipeline and highlighting and affirming Black girls in STEM. In the public health realm, she is involved in community and international health. As a change agent, Shenell's goals are to use her science background to bridge health literacy gaps in the diaspora and to help empower or become self sustainable colonized areas of the diaspora. by eliminating their need to rely on the paternalistic relationships of NGOs and other agencies for their medical or health needs. This includes helping to improve current health infrastructure, increase the capacity to tackle the infectious diseases that were introduced to these areas, and reduce the prevalence of non-communicable diseases, and encourage STEM in public health education so that these services can continue without foreign intervention. So I think those are really important topics and really important work that you guys should know about, because a lot of times STEM looks one way, but Dr. Tolson is definitely an example of how people who look like you and me are doing really interesting and cool things to advance STEM or science research. So while we are waiting for Dr. Tolson, I would like to share with you guys that if this is your first time tuning in to our Teen Science Cafe, we host these every month, and you can catch these twice a month, except for the holidays. And for the month of November, which is coming up in just a couple of days, we are very excited to have not only our cafe, but also our STEM in the City, which is one of our programs, and our Instagram takeovers, which is one of our programs. And I want you guys to remember that when it comes to STEM, as I just said, there's a certain idea or maybe misconception of STEM looking one way, and STEM can actually look a couple of different ways. And if you have an interest in something, you most likely can find it in STEM. So I invite you all to join us for our upcoming Instagram takeover that will be taking place next week. And for our Instagram takeover, our highlight is I would say if you enjoyed tonight's program, then you're gonna want to tune in to the Instagram takeover, because we have two individuals who are Black women in STEM who are making it look cool, who are showing you that you don't have to show up as one way when you are wanting to do research and experiments in the lab. So with that being said, for next week, our Instagram takeover is going to be with Kayla Bradford, who has some great content on Instagram. She's a Black science chick, and she's a PhD candidate at Meharry Medical College who studies host-parasite interactions. And so that sounds kind of like what host-parasite interactions, if you're not familiar with that area of science, but she actually does a really good job of breaking it down. And so Kayla will be taking over our Instagram next Thursday at 5:30 p.m. And we definitely invite you to take a look at our Instagram page, where she'll be sharing like what exactly led her to doing this, not only academic work, but also the research that she does in STEM, and also more in depth about her work, and also the current trends that exist in her STEM area, which is microbiology. So if you guys like biology, then that would be a good opportunity for you to learn more about what it would look like for your own path into college and career in microbiology. Also, for the month of November, we'll be having a Stim in the City with Coretta Venable, who works at the largest weather and climate agency in this country, in the U.S. And she'll be talking to us about her work on she'll be talking to us about her work in November. And so if you are someone, our STEM in the City program is specifically targeted towards earth and environmental science. So if earth and environmental science is something that you have an interest in, if you like getting outside, if you like doing research as far as plants, and in some cases animals as well, but for this specifically, it will be the environment that we all live in, then I definitely invite you to tune in for our STEM in the City on November 13th with Coretta Venable.
Okay. Well, Carlin, Dr. Tolson is back in, so I'm going to let her in. Okay?
Okay.
Good evening, Dr. Tolson.
Hi. You guys see me, hear me okay?
We can see you and hear you okay. We're so happy that you got your tech issues resolved because you were such a great volunteer. We're like, oh no, we have to hear. The teens need to hear.
I know. I was so worried. I was like, oh my gosh, oh my gosh, oh my gosh. We were already online at first and then crashed. So that was really surprising for me.
Anything could happen, right?
But I'm glad you're back. Yeah.
So Carlin already introduced you. So we are going to go ahead and let you take over the cafe.
Okay, great. So I'm on my cell phone right now. And so while my computer is restarting, I'm going to work from my cell phone and then I will transfer. Once I get in on my computer, if you guys can let me in, that'll be awesome. And that way we can have a full spectrum cafe tonight. Hi, everybody. Welcome to the Teen Science Cafe. My name is Shenell Tolson. I'm a scientist and also a public health expert. My degrees are in biology, molecular biology, and bioinformatics, as well as public health. I'm sure Carlin already told you all that, but I wanted to reiterate so that you understand that I'm a doctor, but not that kind of doctor. But I still know what I'm talking about a lot of the time. I know what I'm talking about. All right, so tonight we're talking about DNA and me. And I know that usually when we talk about DNA, we talk about things like double helix structure and base pairing and things like that. But did you know that DNA is not just your genetic code? I mean, it is, but it does so much more. We could do so much with genetic code. So when we think about genetics, we think about how all the things that make you who you are come from your genes. Right? So as a whole person, as a human being, you have 46 chromosomes. Chromosomes are what make up your genes. You get 23 of those chromosomes from your mom and 23 of those chromosomes from your dad. So that means that one half of your DNA comes from one of your parents and the other half comes from the other of your parents. And to keep, if you can put up that slide, I think it'll be a little bit easier for everyone to see and understand. And I can't see it, so can you let me know when it's up?
Give me a second. I'm bringing it up. Okay. You said the one with the parents, right? That's the one you want to see?
Yes. The one with the parents. Chromosomes, 46 chromosomes.
Is that I think this is it. Give me 1 second. I'm sorry.
Yeah, sure.
I had it.
So watch that up. The part of this is, so you get half your DNA from your mom and half from your dad. There we go. So 23 from dad, 23 from mom equals you. Okay, so we get half from everybody, half from mom, half from dad. So you can be a boy or a girl. Either way, a whole human person has 46 chromosomes. So why is that important? That's important because when we talk about genotype, This is what we mean. And our genotype is what our DNA is made up of, the things that make us who we are. Now, our phenotype is the physical representation of who we are. So what we look like, what color hair we have, what color eyes we have, how tall we are, all those things go into what make us who we are. So a genotype is the genetic code that makes us who we are. And our phenotype is how those genes are expressed, how we look. Does that make sense? Okay, hit me in the comments so I can, so Takiwa and Carlin can see what you're saying, and we can ask questions and go back and forth. I want this to be very, very interactive. Okay, so now based on that, let me ask you a question with the understanding of what a genotype is and the phenotype is. Takiwa, can you put up the mice cross, please? In this picture, you're going to see two mice, and we want you to be able to look at those two mice and make a decision about what kind of offspring you think those mice will produce. Okay, great. So you see one black mouse plus another black mouse. So in the comments, can you let Takiwa and Carlin know what kind of mouse you think, what kind of offspring or mice babies, what color will they be? Can you put that in the comments? We want to know what you think. I'm going to peek in the chat and see what you guys are saying.
Okay, so Dr. Tolson, I have a question.
Yes.
And maybe somebody else is thinking this. So if you have this question to put in the chat, the mice look is almost exactly the same. So in that case,
so their phenotype is the same.
Okay. So is it connected to what you said? And I'm sorry to interrupt. I'm like, how are we going to figure this out? They look the same.
That's the whole purpose. That's a very good question, Takiwa, and that's the whole purpose. So just by looking at the phenotype, it's pretty hard to guess what kind of offspring will come from these two mice, right?
Mm-hmm.
Because we don't know their genotype. So that's a very good question, and that's the entire purpose of looking at these two mice. So we see one black mouse plus one black mouse. Phenotypically, what they look like, you would think that all the mice will be black mice. Does that sound about right? Okay, great. Can we go to the next slide that I sent you, which is the actual Punnett Square of the two mice? And while she's pulling that up, the purpose of this is to show you that just by looking at something, you can't tell what the genetics are. And so here's what we call a Punnett Square. A Punnett Square was used by Mendel to show inheritance, genetic inheritance. And so here at the top, we have the mom mouse at the top, and the genotype is big B and little b. The dad is on the left-hand side, which is also a black mouse, and dad's genotype is also big B, little b. Now, big B means black in color. Little b means brown in color. So even though these two mice are both black, as you can see, they both have a little b in their genotype. That means that even though these mice are black, they carry the brown color gene. Does that make sense? Now, as you move forward, each of these 4 square blocks represents one offspring. This is called a probability, right? So out of 4 offspring, here's a probability of their coat color. So on the first square, the top left square, remember you get one inherited set of genetics from your mom and one inherited set of genetics from your dad. If mom gives one of her big B's and dad gives one of his big B's, the first child will have the genetic genotype big B, big B. In the second square, or the top right square, mom gives her half of genetics, which is the little b, and dad gives his big B. So that's the other half. So that child number 2, will have big B, little b. Moving down, the bottom left square, mom gives her big B, that's her half contribution. Dad gives his little b, that's his half contribution. So that child will also be big B, little b. And then we move over to all on the right, on the far right at the bottom. Mom gives her half, little b, and dad gives his half, little b as well. That child will end up getting little b, little b. Now again, we started off with 2 black mice, but those black mice were only black in phenotype. In genotype, they were black mice that carried the brown mouse trait. So if you move over to the right of the diagram, you'll see all 4 offspring represented genotypically and phenotypically. Offspring 1 is big B, big B. That's a black mouse. Offsprings 2 and 3 are big B and little b. Those big B's mean that's another black mouse. But the 4th offspring, the 4th baby mouse, is a little b, because again, you get half your genetics from your mom and half from your dad. And so the 4th mouse, this is probability, not guaranteed, but high probability, the 4th mouse is going to be a brown mouse. And that's how genetics plays a role in how we look and how we pass on genetics. I hope that made sense. Did you have any questions in the chat? Okay, good.
I don't have any questions. It's a quiet bunch.
It's a quiet, right?
It's a quiet bunch tonight. So maybe they just know genetics really well, or you're explaining it so good.
Well, that's good. That's always a good thing. We aim so that people can understand. All right. So now that we have that taken care of, Punnett squares help you understand how genetics are inherited and how they move from generation to generation. Okay. Once we know what the genotype is, we're able to predict all the possible phenotypic outcomes, what things or organisms will look like. Now, you have the unique set of DNA. You have half from your mom, half from your dad. There's literally nobody else in the world like you. Even if you had an identical twin, you still wouldn't be exactly alike. Although your genetics would be alike, you know what's different about twins? Their fingerprints. So you are uniquely made. There is nobody else in the entire world that is just like you. Now remember, you get half from mom and half from dad. And genetics are not just what we use to predict phenotypes. We can also use it to predict things like inheriting diseases. So for example, sometimes when we inherit genes, they're not always good genes. Sometimes there are genes that can make us sick. Those genes are called mutated genes or mutated DNA. And that can happen one of 2 ways. One, it happens because it's a mutated gene within your family lineage and you get passed on from generation to generation. Or it could be an outside factor, we call it an environmental factor, that can mutate your DNA and lead to genetic changes. So now, we just did the experiment of showing you phenotype and those phenotypic changes. What if we moved forward and talked about diseases? All right, so let's talk about one that's really popular in our community. So if you're Black in America, you have lineage to Black people in Africa, the continent. And how do we know this? Because our genes tell us so. In Africa, having a certain trait called sickle cell is useful for protection against diseases like malaria that can affect the blood. Now, the organism that passes on malaria lives inside of mosquitoes. So if you get bit by a mosquito and that mosquito is carrying the parasite, it can pass to you through a bite in the skin. Now, once that happens, the parasite will attack healthy red blood cells, and then you'll start to get all kinds of sickness happening to your body. Now, if you have sickle cell and you get bit by a mosquito that carries parasite, you won't get as sick because the parasite needs a big, fat, healthy red blood cell to survive. A sickled cell is a more shriveled up, deformed red blood cell. It's not really a good thing for us in the US, but again, if you are of African lineage, you may have that trait. it's an advantageous mutation if you live in that part of the world. Okay, so now the only way you can know if you have sickle cell or the trait is through genetic testing, similar to what we just showed in the last Punnett Square. You can't tell by looking at someone, their phenotype, if they have it or if you have it. The interesting thing about genetics also is that you don't have to have the full-blown disease to pass on the genetic trait. So let's use that Punnett Square example again to see how this works out. So Takiwa's going to pull up the Punnett Square of two parents that have sickle cell trait. And we're going to see, using the same Punnett Square example, how genetics are passed on from mom and dad and can lead to either disease, no disease, or traits. Okay, so we can see clearly here, we have a mom at the top, and mom has, okay, can you hear me now? 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 Okay, can you hear me now?
So your computer came on and it was still in the stream. So which do you want to go to your computer? So that's the echo. Okay. It's going to remove your cell phone and put you back on your computer. Okay. Okay.
Okay, did that work?
That works. Okay.
Awesome. Okay, we're back. Okay, we're back. All right, so as you can see at the top here, we have mom. And mom has a big S and a little s. And then on the left-hand side, we have dad. Dad also has a big S and little s. Neither mom nor dad have sickle cell disease. Big S means you have regular blood cells. And little s means you have sickled blood cells. So neither one of them have the actual disease, but they do have the trait. So when we go through the same process and we look at the contributions that mom and dad give, in our first square, the top left, we get a big S from mom and a big S from dad. In the top right square, we get a little s from mom and a big S from dad. At the bottom left, we get a big S from mom and a little s from dad. And in the far right corner at the bottom, we get a little s from mom and a little s from dad. So it's the same process that we did with the mice. After 4 children, this is probability, some of the children will have the full-blown sickle cell disease, little s. Some of the children will have no disease and no trait, big S. And then some of the children will have no disease, but they will carry the trait for sickle cell, just like their parents. And that's offspring 2 and 3. So this is how we can look at how genes are passed on from generation to generation, and how we can look at genotypes to determine the outcomes of the passing of disease and health. Any questions there?
So I have a question.
Yes.
So occasionally I donate blood, but last year I figured the Red Cross told me that my blood is good for sickle cell patients. But as far as I know, I don't have sickle cell disease. So, but I was curious, like, okay, what is in my trait that makes the blood good for sickle cell patients?
Now that I wouldn't be able to answer right out, but I can definitely ask a colleague and get back to you on that behind the scenes. That's very
okay. Is that something that, like, maybe it may be recessive? Just curiosity. I mean, yeah, you can tell me later, but I was just curious.
So yes,
I've been having
sickle cell is definitely, it's a recessive trait. It is recessive, and so this is also why, if you look at the photo we have here, because the little s and the big S are together, but you still end up not having full-blown disease, that's how we can tell that the little s or sickle cell is a recessive trait. So it's not dominant. Okay. And that's also how you have to have 2 little s's in order for someone to actually have full-blown sickle cell disease. Also, good question. All right, so that's how you can have a mutated DNA or have genetic traits that are passed on with disease through genetic inheritance happen from generation to generation. But I also mentioned beforehand that there's also environmental factors that can cause genetic changes. So let's take a look at that. Have you ever heard about endocrine disruptor? An endocrine disruptor is something that's going to cause a problem with how we have hormones in our body move through a system. And so typically hormones regulate different functions of our glands, of our organs, of our organ systems. So think things like puberty, think things like digestion, think things like being able to have milk, things like that. So there's hormones and enzymes that are regulated by your hormones and your glands. But if you have a disruption in those things, you won't be able to move forward and use them appropriately. So for example, someone who's diabetic, they don't make the hormone insulin. Or if they do make the hormone insulin, their body can't receive it and accept it the way that it needs to. Now, if that happens, then you aren't able to eat food and regulate your own blood sugar. That's how that hormone works on that particular process. And we have hormones for different processes for all across our body. If you have a disruptor that you are exposed to, whether it be by air, by water, by soil, maybe by food, it can disrupt your entire system. And that can cause genetic changes. Those are also mutations, but those are not passed down by inheritance. They are what we call acquired or environmental stressors. And there's a short video clip that I have. It's about 3 minutes long that I wanted to show you. And as soon as Taki was able to pull it up, we can go ahead and watch that really quickly. It's a very short video, but it really explains in further detail how endocrine disruptors affect our bodies. And that's how they make genetic changes that aren't necessarily good for us. It can harm us in the long run. And while she's pulling that up, I want to give you some fun facts. So, did you know that we just mapped the entire human genome last year? So what that means is we know the genetic code for every trait that humans have. So that means that whatever disease, whatever trait, whatever nuance in passing things down that could ever be known, we know it now. And it took a really, really long time to figure that out. There were lots and lots and lots of scientists who worked together on this, and it took about 50 years. Now, here's another fun fact. If you are a really fast typer, and I mean like 60 words per minute, if you could type 60 words per minute for 8 hours a day, an entire workday, it would still take you about 45 to 50 years to type up the complete sequence of the human genome. It's huge. That's how big it is. Something else that I bet you didn't know either. There are actually 5 different types of DNA. There's B DNA, there's Z DNA, A DNA, C DNA, and D DNA. But the kind that we're used to, double helix kind, that's going to program us, that's the B DNA.
Okay, I have the video up. You ready?
Yes, we're ready. Thank you.
If you've been listening to the news at all, you've probably heard about the dangerous chemicals that are called endocrine disrupting chemicals, or EDCs. Inside our bodies is a network of glands and organs called the endocrine system that produce, store, and secrete hormones. Normally, the hormones go from the gland or organ to the other organs and fit right into specific receptors expressed in cells. Kind of like how a key goes into a lock. But when EDCs get into the body, they can mimic the shape of the hormone, and they go into the receptor instead. They can trick the body into thinking they are a hormone or block the hormone from fitting into its receptor. Fooling a cell in this way can trigger abnormal processes in the body. This can wreak havoc on the entire endocrine system and may lead to severe health consequences. EDCs have been linked to human health issues related to sperm quality, fertility, abnormalities in sex organs, endometriosis, early puberty, nervous system function, immune function, cancers, breathing problems, metabolic issues, obesity, heart health, growth, neurological and learning disabilities, and more, because hormones regulate nearly every process in the body. What's worrisome is that exposure to EDCs can happen anywhere and come from the air we breathe, the food we eat, and the water we drink, and through the skin. Some EDCs even transfer from mothers to fetuses and infants. They can be found in industrial chemicals that leach into soil and groundwater and then make their way into the food chain and build up in fish, animals, and people. Consumer products such as plastics, household chemicals, fabrics treated with flame retardants, cosmetics, lotions, products with fragrance, and antibacterial soaps, pesticides, fungicides, or industrial chemicals in the workplace. The best way to avoid exposure is to check labels and avoid products containing known EDCs. Research on EDCs is growing, so watch for new information on products to help your family's health. Contact your elected officials and tell them to pass the Personal Care Products Safety Act, which would create an FDA review process to check the safety of ingredients used in personal care products like toothpaste, soap, and sunscreen. Patients have questions. We have answers. The Hormone Health Network is your trusted source for endocrine patient education. Our free online resources are available at hormone.org.
Okay, great. Thank you. So now that we know about how genetics are passed down for generation, we know that there can be mutations that can be passed down for generation or due to environmental factors. Did you know we can talk about extracting DNA later on? That later on is right now. So we're going to extract some DNA. It's really easy to do. And if your parents allow you to, you can get the ingredients. I'll give them to Ms. Takiwa, and she will have them posted for you all. And you'll be able to extract your own DNA at home safely with your parents. So let's go over our ingredients. First off, because we're scientists, we got to have our gloves on and our goggles, okay? So I'm prepped, okay? I'm not in a real lab, but it's my little makeshift lab. But we also have safety first, okay? So my gloves are on and my goggles are on. Here are my ingredients. I have some strawberries, which is where we're going to pull our DNA from. I have some liquid dish soap. I have some salts. I have some water, just plain water. And I have some isopropyl alcohol that has been chilling on ice. Yeah, it's cold. Chilling on ice while we've been having our conversation. I'm going to put it back here on ice until we get ready for it. All right, so are you ready to extract some DNA? All right, so the first thing we need to do is take about a tablespoon of our soap and add it to the water. And I think that's about a tablespoon. Okay. Good. And then we want to take about a teaspoon of our salts and add that to the water too. So that is about a teaspoon. Okay, so now we're going to take our baggie and we're just going to shake, shake, shake. So we shake, 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 shake. Shake our experiment. Okay, now what we're going to do is we're going to take our strawberries and we're going to dunk them in the bag. Just like that. Strawberries are plants. They're living things. So believe it or not, they have DNA too. So now we're just going to take our fingers and we're going to smush our strawberries in our solution. Okay. Don't forget to smush them, smash them really good. We want to make sure we get all those little bits nice and mushy. Now, why are we doing this? Because we want to break up the cells. So DNA is found inside of our cellular material. And so the way that we are able to extract the DNA is to break open the cells. So the salts and the soap are helping to break open these strawberry cells. And once they're open really good and the DNA is accessible, it's going to be in this liquid. Okay, so we got it all nice and smushed. See how the color of the water changed? Really good stuff. All right, so, I have to mention, I also have tea bags. So you can use a coffee filter to do this, but I'm not a coffee drinker. I'm a tea drinker. So I have tea bags. So what we're going to do is we're now going to take our strawberry water that has all the open strawberry cells in it. We're going to take it and we're going to filter it to get all the chunky bits out. We don't want any chunks in our final solution. And here's my dish that I'm going to pour it in, okay? Here's my dish. And so I'm going to pour the solution. It's so soapy. There we go. I'm going to pour the solution into our filter. So all we're getting is just the liquid. We're not getting any of, can you see how the liquid's pouring out? We're not getting any of those strawberry bits. We're just getting the liquid. And that's it. Just the liquid. Okay. And wipe myself up a little bit. Paper towels. So we want a clean lab space. And so now, as you can see, what we have here is just the liquid. There's no chunks in here at all. Okay? Now, here is the fun part. We're going to make this move a little faster. We are going to take our alcohol that was on ice, so it's nice and cold. I'm dumping this out a little bit just so we have a little less, so it'll move faster for you. And we're going to take the alcohol mixture and we're going to pour it in really slowly. We don't want to disturb any of the bits. So we pour it really, really slowly. And we're going to watch this reaction. Now, you can't see it, but I can see it already. That DNA is starting to come out of these cells and separate. So once I finish pouring in the alcohol, I'm going to show you the layers that are forming. Okay, good. Now we're going to let it sit for a minute. Okay, so here's what it looks like. I'm just going to show you what it looks like. See the two layers? Okay, good. So we're going to let this sit for a minute, and I'm going to time it exactly. I'm going to put my timer on right now. I already had it set for you guys. And so we are counting down right now, okay? And so once this goes off, we'll be ready to extract our DNA. And it's already happening. So now while this is taking place, let's talk about living things. So living things all have DNA, whether you're a plant, you're an animal, you're a reptile, you're a dog, you're a cat, a turtle, everything that's living has DNA. And so you can do this with lots of other things. Why did I choose strawberries today? I chose strawberries because they're pretty easy to do. They're pretty easy to smush up. They're pretty easy to disassociate, break open the cells, and they're pretty easy to extract the DNA from. But you can do this with just about anything as long as you have the right materials, okay? We're almost done. We're about halfway through. We're almost done letting this sit and letting the DNA come out. Okay, we're done. So I'm going to have my trusty stirring rod. I'm going to stand up and show you this. Here is the DNA that's coming up. Actually, let me use a toothpick. It looks really, really slimy like this. This is your DNA, guys. This slimy stuff, this is DNA. This is what DNA looks like when it's extracted. Pretty cool, right? So I'm going to put this back in here because we don't really need it anymore. But that's how you extract DNA. Wasn't that neat? Anybody have any questions? Hi, Carlin.
Hi, I was trying to unmute myself. I love how this slimy is perfect for the season that we're in.
Right? It's a great little thing to do at home with your parents with permission. But it's pretty cool. It's pretty easy. It's really easy to do. And again, it's real DNA. This is plant DNA, strawberry DNA.
So is that what human DNA is going to look exactly like?
Well, if you were to extract it that way, it would definitely be slimy. Yes.
I love that. That is very good to know.
That's pretty cool, isn't it?
I think it was. Yeah. I definitely think that was cool. And I think that it's so great that the kids that are watching will be able to do it at home as well, and we'll send them the ingredients and the steps so that they can experience it for themselves as well.
Yeah.
So is it Q&A time?
It's Q&A time.
Okay. So I definitely wanted to ask, one of the main things that we focus on with STEMlink is the exposure to different types of scientists. And I think that, or not even I think, in the STEM field, it is said that it is so important that that sort of representation exists. So what led you to being interested in STEM? Did you have examples of STEM growing up that kind of led you to this path, or what was your career journey into this?
So I didn't really have many direct examples of STEM or people that were in STEM that look like me or around me. I grew up on Bill Nye the Science Guy. So mostly when I got STEM from, my STEM interactions were from TV, So I'm a Bill Nye kid. And so things like that are what really got me into science. But as far as when I got older, but when I was a little itty bitty thing, I've always been interested in science. It's been so much fun, and playing outside and picking things up and looking at things, streams and frogs and turtles, all that kind of stuff. So I've always had that in me. And I also had a little doctor's bag. And so I would pretend when people were sick, they got an owie, I would give them medicine and check them out. So I've always been like STEM oriented, even from a little kid. And then, my parents, they saw it and they were like, oh, okay. I mean, if she likes it, I was like a weird little kid. So if she likes it, like, okay, keep going.
Right. Yeah.
Yeah.
I love that. And then as far as what you do every day as a scientist, because I think one of the most interesting things to me about the STEM field right now is that there are a couple of different ways that you can reach different maybe non-scientific audiences with awesome STEM research and STEM work. So what do you do every day as a STEM professional?
So what I do every day is I am a biologist. I'm trying to think of how to say this in latent terms. So I'm a biologist. I work in toxicology. And so what I do is I make sure that things, or I assess things, the risk or the hazard associated with different chemicals, if they're safe for humans and for the environment. So right now, I used to do biochemicals, so things that are naturally occurring, like lemon oils and lavenders and things like that. So things that are naturally occurring. Now I'm on the conventional side, so synthetic materials, but I work with dietary and ORE. So ORE stands for occupational and residential exposures. So I work in exposures now. So when we're exposed to different things, I assess those things to see how safe they are for us to be exposed to.
And that is so important right now, right? Where we're kind of in the STEM community, and particularly when we talk about being environmentally sustainable, how important it is to kind of take a look at, as you said, like the things that are not good for our bodies and the environment, and the things that are okay for our bodies and the environment.
So the things that are good for the environment and not good for the environment are things that we look at by assessing the test. So someone will do a test in their lab. So I don't like to work in a lab. I used to, but now I don't. I'm on the other side now. So instead of doing the lab work myself, I look at their research. So they will perform the experiment and they will send me the data. And I'll look at the data and determine if, and I will translate that data into what we call layman's terms or real life language and let someone know what that means. So I extrapolate data. So if the numbers are this, and that means bad, then I say, hey, this is bad for the environment or bad for our health. If the numbers are that, I could say this is okay, it's small amounts. And so it's not just saying good or bad, it's also about understanding the amounts that we're exposed to as well. Being exposed to anything for a certain period of time is never good for you, right? Anything. Mm-hmm. So in toxicology, we say that death is in a dosage. So it's not really about what it is all the time. It's about how much of it is. So there's certain things, of course, that tiny amounts will harm you. But there are other things where as long as you don't have lots and lots and lots of it, it'll be okay.
This is an interesting comment that someone made. They said that they saw another video where someone extracted DNA from a strawberry, shrimp, and kiwi. But they said that the kiwi's DNA was really small. And then they also said something about the taste of DNA. Could you speak to, are there different variations in how big or small the DNA is, and if there are other aspects of it as far as sight, taste?
So let's talk about that. So earlier we were talking, I told you that if you're a human being, you have 46 chromosomes. You get 23 from mom and 23 from dad. So half of 23 is half, and 23, the other half equals 46. You as a whole person. But that's human DNA. So other things don't have the same number of chromosomes. So different organisms carry different number of chromosomes, and therefore their DNA looks different or will be different. And so I think that's what that person's referring to.
Okay. And so another thing that we were curious about was what made you decide to go to graduate school? Because, as all of us who have been watching this presentation, like we are, as you said in this presentation, you're a doctor, but you're not that kind of doctor. Yeah. And so for a lot of STEM professionals, a lot of scientists, it is understood that they do go that academic route as far as going all the way to getting their PhD. So can you just talk about what made you decide to go to graduate school and get those credentials?
Yeah, sure. So as you mentioned earlier, my bachelor's degrees are in the hard sciences, so molecular biology, biochemistry, etc. And so I'm a scientist. As I moved up, I think it's super important. I work in the community a lot. I do things like this, work with SEMLEAK a lot. So I do things where I need to be able to talk to people who are not scientists and relay important information to them in a way that they can understand and they can absorb. And so for me, what that looks like is, and with community work, I work with helping people understand communicable diseases. So things like infectious diseases, like STDs and things you can get, like COVID, common colds, etc., and phenomena like that. I also work with people and talk to them in communities about non-communicable diseases, like heart disease, like diabetes, like high cholesterol, things like that. But in order to do that, I think it's super important to use STEM in order to be able to relay information to them that will help them make positive behavior changes. So, for example, I think that if people knew how diabetes and blood sugar worked, they would be more inclined to change their behaviors versus going cold turkey or having to be told go cold turkey and stop eating certain foods and then be non-compliant. And what I found is when I explain using a STEM concept, like, hey, whether you eat a carrot or a cupcake, all food breaks down to sugar, because not many people know that. So it doesn't matter what you eat. Cars run on gas. You want your car to go, you put gas in your car. You want your body to go, you need to eat food, because food is our fuel. Our body's fuel is converted into sugar called glucose. So it doesn't matter what you eat, whether it's a carrot or a cupcake, it's all sugar. So again, it's not about necessarily what you eat, it's how much of it you eat. And so understanding those kind of concepts, and again, I bring STEM into that, understanding that people are like, oh, and they're able to better cope with the diagnosis that they get. And they're also able to make different behavioral changes because it's easier for them to make those changes instead of cold turkey, stop eating certain foods. Does that make sense?
I think it does.
Okay,
Yeah.
Good. So that's why I wanted to go to grad school, because I wanted to be able to work in the community, but also I wanted to be able to be an authority in that segment. So the difference between a DRPH and a PhD is someone who gets a PhD will probably, in my field, will probably want to do academics. If you have a DRPH, you're more of a practitioner, so you practice. And so for me, going the grad school route was about using my science, my STEM, to inform healthy communities and public health.
Yeah, no, I think that makes perfect sense because like I am not from a STEM background, so I think that a lot of times STEM research gets lost in translation, where it's kind of like, okay, well, how does this apply to real life? How is this relevant? But what you're saying, you do need those people who can break it down and show and tell how it is applicable to other people or everyone really in the world.
Yeah. And then we call that science communication, basically.
Yes. Thank you so much for pointing that out.
Yeah.
Well, that was our last question for this cafe. Were there any parting words that you wanted to share before we wrap up?
Well, it was really fun. I really enjoyed this. I always like working with STEMLink, so there's always great projects and programming. So it's really fun. I hope that everyone got something from this Science Cafe. I'm so sorry that we had technical difficulties. There was so much to talk about, but hopefully with what we were able to talk about, you did take something away from it. You learned something, and you'll be back for the next Teen Science Cafe.
Yeah, and thank you so much, Dr. Tolson. We love you. You are a multi-volunteer. So I was just like, oh, she's doing this cafe. Then Carla reminded me of your IG takeover, and I cannot wait until it is safe for us to do in-person program in the DMV again. Because I was looking at pictures the other day of you with the babies. And so thank you so much for giving of your time, for teaching us about DNA and I can never remember how to say it, but the square.
The Punnett Square.
Yes. Okay, that's the only thing of biology I understood because I have a bad memory, so I can't do biology. And before we go, I just want to make everybody aware of Carlin put a survey in the chat. And so please take a few minutes to complete the survey and allow us to get your input on this cafe. what you learned and what you may want for future topics. And so thank you everyone for attending. Thank you so much, Dr. Tolson, for giving of your time and doing another great presentation to make STEM fun and exciting, and for always having bomb science gear. I think one of the things I love about you is you just show this, you totally break all the stereotypes, right, of what scientists should be, right? And we know what they say that they look like, right?
Yes.
But even you have a scientist wearing a scientist t-shirt, and some of the things I see on your Instagram page, I need to order them, but I always like, okay, let me not make this impulse buy, but I need to order that. So let me think about it in a few days. So that's what I think I am going to order. So thank you so much. Thank you everyone for us joining the cafe, whether it's live or you're watching it on our YouTube channel. And we hope you continue to learn about STEM careers. And we hope that if you were interested in pursuing being a public scientist, being a molecular biologist, and a biochemist, that Dr. Tolson gave you some tips and advice on how to pursue that path and gave you an idea of what those scientists do. And thank you so much, everyone, and have a great rest of your evening.
Bye, everybody.
Bye.